The process continues in a cascade with more and more genes affected. The end result? A body part, and the answer to how one part can think it's another. A mutation causes a cascade in the wrong place, a leg where an antenna should be. Only about 12 genes out of around 15,000 in the fly are the master architects of its shape, telling the developing organism where to form a leg, a wing, an eye. A remarkable discovery that only gets stranger.
These observations were essentially all worked out in flies, and with little or no expectation that they'd apply very directly to other animals. However, we now know that in fact these genes, these same homeotic genes, exist in most or all animals, at least all reasonably complex ones. And these genes are used in most animals to control where along the head-to-tail body axis different structures will be formed.
The work of Matthew Scott and others has uncovered a set of master architect genes that perform similar functions in almost all animals. Thanks to flies and our shared genes, scientists are abuzz about the possibilities for bold new discoveries in humans.
Where we can start with the economy of scale of a fruit fly and do our genetic work, which almost inevitably now will lead to significant insight to the biological underpinnings of similar functions in humans.
At Cold Spring Harbor Laboratory in New York, Jerry Yin, a molecular biologist, and Tim Tully, a behavior geneticist, have taken the study of fruit flies from body structure to behavior, trying to understand one of science's great mysteries, memory.
Memories are so personal. They really are a huge component of our self-identity. You know, the memories that we carry around, the memories of all sorts of things really, to a large degree, constitute us.
But how do you test the memory of a fly? That's Tim Tully's job. He's the Pavlov of flies. Using mild, discomforting electric shocks paired with distinctive odors, he teaches flies to run a maze.
And I've trapped them in there, in their training chamber. I'm going to apply vacuum to draw air through it and hook the electric current up to the floor of the training chamber. And now to begin training, I'm going to put one odor on the end of the training tube. This is octanol that smells a little bit like licorice. And I'm going to turn the current on. So now air is being drawn through the top of this chamber, out the bottom, and through the training chamber. And the flies are being shocked for 1 second every 5 seconds for a total of 60 seconds of training. After 45 seconds, we're going to put a second odorant on the training chamber, but this time don't turn the shock on. So they've been shocked in the presence of octanol, not shocked in the presence of methyl cyclohexanol, which smells like my tennis shoes in July.
The flies are lowered in an elevator to a tea maze. On the left, the shocking licorice smell. On the right, the safe, smelly sneakers. Most of the flies avoid the smell associated with shock. With 10 training sessions like this, an average fly will create a long-term memory to avoid licorice smells. With the test in place, all Tully needed were the right subjects. And that was Jerry Yin's job. Before coming to Cold Spring Harbor, he had isolated the gene most likely involved in long-term memory formation in flies. It's called the Krebs gene. It seemed to produce two products, the Krebs on switch, which increased gene activity, and the Krebs off switch, which suppressed it. To test the gene, Yin genetically engineered flies that would only make the Krebs off switch. When Tully tried to train them in the maze, the results were shocking.
We saw that the appearance of long-term memory itself was disrupted. No other aspect of the fly was affected. It still smelled the odors normally, it still sensed and escaped from the shock normally. It just couldn't form a long-term memory of that event.
The genetically altered fly could not be trained.
This is a very encouraging experiment, but it's not absolute proof because you might be disrupting this process in a very indirect sort of way.
Intrigued, Tully and Yin next tested flies that only made the Krebs on switch.
They actually had enhanced long-term memory. They formed long-term memory after only 1 practice session, when a normal fly requires 10 practice sessions to form a long-term memory. So what expressing the Krebs on switch basically did is it created a fly with the functional equivalent of a photographic memory. Since we use odors and shock, I guess we would have to call it an odorgraphic memory.
Odorgraphic flies? A new flea circus? Perhaps much more. With the shared genes in flies and humans, is the manipulation of human memory far behind?
I think the speculation is both exciting and scary, because certainly what we're talking about here is that memory is a biological process that we can tinker with.
Control over memory raises scary specters. But imagine a drug based on Yin and Tully's work that helps those with deteriorating memories, like people with Alzheimer's disease.
If we can turn the Krebs switch on and essentially increase the gain in long-term memory formation, then Alzheimer's patients that have trouble consolidating short-term memory into long-term memory will get more into long-term memory, and they won't suffer the same kinds of memory losses that are associated with the progression of the disease.
But could there ever be a time when it is better to forget? Tully imagines a long-term memory suppressor as a morning after pill for traumatic events.
Even though one traumatic event can be consolidated into long-term memory, it still takes time after the event to accomplish the consolidation. So if a long-term memory suppressor can be administered in the hospital after the trauma within that critical period of time when long-term memory is being formed, then you can block the formation of that very emotional, traumatic memory.
Strange and exciting possibilities, as revealed by the well-trained fly. They sound like science fiction. Does this mean all we are giant flies?
We are by no means big flies. We are very, very different from flies. And yet, the machinery that creates us has a lot in common with the machinery that builds a fly. So we're at really the opening of a time when we're just beginning to understand how these genes work in any organism.
Our shared genes are a map to the future that is only beginning to unfold. In human genetics, there are few things more fascinating than identical twins. Even now, the biology behind the phenomenon is not very well understood, but the results are naturally intriguing.
I'm Kurt Froman, and I'm Kyle Froman. I'm 3 minutes older, and we're identical twins.
And we're both students at the School of American Ballet, and we're both apprentices with New York City Ballet.
Welcome to Twins Restaurant in New York City. I'm Debbie Gans. I'm Lisa Gans. We have 37 sets of identical twins that work here. And if one gets sick, they both stay home. And if one's fired, they're both fired.
I can never imagine, not being a twin. People ask me, what's it like being a twin? And, I say, what's it like not being a twin? Because, I've never had to, even spend a day without somebody, looking exactly like me, right beside me.
Like everyone who's single always says, I wonder what it's like to have a twin. And we always well, what if there are 3 of us?
Yeah, Kyle is, he's much more organized than I am. He's always got everything in the right place. And Kurt's more of the slob, I'm the clean freak.
It's like having your most annoying quality standing next to you for the rest of your life. The quality. We call it like being born with your soulmate. With your soulmate.
Like 1st or 2nd grade, we had the little cowboy belts with the name, like, you know, across the back. And so, like, April Fool's Day, we'd usually, like, switch belts, but it would never work because we could never
Nobody would even care.
I know.
Oh, we've switched dates, we've switched classes. And I also actually, I had a boyfriend and someone asked me out and I told him I was Lisa because I thought he was cute and I knew she'd like him, and then he went out with Lisa and never knew. He never knew the difference.
If we had been, like, separated when we were born, I don't know, it's really strange because I see these things on TV about twins, like, you know, that they were separated and how
they still have the same taste.
I think that if we were separated at birth, I think we would look alike and, we'd be similar. Maybe. I'd probably be a little more conservative.
Not noticeably to you, maybe on the outside. Maybe my skirt's an inch shorter, but you can't get much shorter than they are to begin with.
We're both in New York.
I don't know if that's just in your personality, like,
I guess
it's just in, like, the way you're brought up. I'm not sure about that.
Genetic?
Twins come in 2 varieties, fraternal and identical. Fraternal twins are the result of 2 fertilized eggs, and that's why they only average the same amount of shared genes as any pair of brothers or sisters. Identical twins are both produced from a single fertilized egg, and that's why they have identical genes. To some, twins provide a natural laboratory to study the role of genetics in human behavior. Here, at the University of Minnesota, in the Twin Cities no less, is the Minnesota Center for Twin and Adoption Research, with an international registry of over 5,000 pairs of twins. By studying genetically identical twins, especially those raised in different environments, scientists here attempt to separate the influence of genetics from environment on human behavior.
The Minnesota study of twins reared apart is a long-term medical and psychological study of identical and fraternal twins who were separated very, very early in life and reunited in adulthood. The purpose of the study is to answer the classic nature-nurture question: to what extent is variation on these traits due to genetic factors, and to what extent is it due to environmental factors?
It may seem surprising that there are even enough twins raised apart to study, but by drawing from a large international population, the project now has over 100 pairs. The most common cause of separation is divorce or death of a parent. In some cases, twins have been adopted into separate homes. That's what happened to Carol and Sylvia, identical twins who were separated at birth. They grew up in neighboring towns in England, but never met.
Now, did you know you were a twin at that point in time?
Yes.
You did, so did Carol.
At the age of 23, Sylvia moved to Australia, making it unlikely she would ever meet her twin. But a chance encounter with a mutual acquaintance brought the two together in their early 30s.
I met a friend who I hadn't seen for many years, who had a friend with her. And it just happened that that friend was a friend of Sylvia's and still wrote to her in Australia. And so, you know, she gave me Sylvia's address and we started writing.
I think I read the letter about 6 times before it sunk in. And then I was going around telling everybody.
Do you feel more complete knowing your twin?
Yes, yes, definitely. There was always something missing.
The twins wrote each other for 9 years. Then, within months of their first face-to-face meeting in Australia, they agreed to enter the Minnesota Twin Study. The study puts the twins through a battery of tests. By comparing the results, Bouchard and his team hope to determine to what degree aspects of personality and physical makeup are genetically influenced. At the university hospital, the twins are literally put through their paces during stress tests.
You let us know if you're going to have to stop, all right?
It is easy to imagine that a person's genes are influential in areas like height, weight, or reaction time. But what about apparently non-biological traits, such as religiosity or job interests? The results from twin studies are both controversial and surprising.
The general findings are that genetic factors influence virtually every psychological trait that we can measure reliably. There's a little bit of variation. IQ tends to be a little more heavily genetically influenced than personality. Vocational interests seem to be a little less genetically influenced, but these things average around 50%. So about 50% of the variation seems to be under genetic influence.
Perhaps the most startling conclusion is that twins like Carol and Sylvia, who were reared apart, are as much alike as twins reared together, like the Fromans.
Common family environmental influence is not a big factor. That is, that the similarity between twins in a family is largely genetic. Now, families are important in the sense that they actually cause some of the differences between twins. Even identical twins who grow up in the same family are only about 50% similar. That means they're about 50% different.
Like, just from meeting us right now, you probably think, oh, I mean, I don't see any difference really, but, you know, if you're around us every day, then you see, you know, who's more, you know, quiet.
All of a sudden, she doesn't like to fly, so sitting on a plane for 20 hours, and I'm snoring, and then all of a sudden you hit turbulence, and she goes, "You feel that?" Yeah, I felt it because she just punched me in the arm, and she woke me up.
I'm a little bit more easygoing, you know, I just take things as they come, and Kyle is the one who, like, sits there with a calendar and says, oh my God, we gotta do this and this and this.
The Minnesota twin studies document both the differences and similarities between twins. But it is the similarities, especially the quirks like Carol and Sylvia's shared taste in food, that capture the imagination.
If you study identical twins, you find remarkable idiosyncratic similarities, things like, well, they bite their nails and they bite them to the same extent. With twins reared together, you can explain it, of course, or explain it away by saying, well, they had similar experiences and it caused them to have these likenesses. With twins reared apart, it's a lot harder to do that. It's our feeling that clearly there are genetic influences on all kinds of idiosyncratic behaviors, but it's a hypothesis that's almost impossible to prove.
Does that smell most like lemon, chocolate, root beer?
The similar habits, gestures, and personalities of twins raise fundamental questions about human nature and the role of science and society. Some fear that work like Bouchard's can hurt the disadvantaged by suggesting that they were born that way. It's in their genes. But Bouchard is careful to qualify his work.
We should appreciate that all of these twins have grown up in standard middle-class environments. Some in environments a little better, some in environments a little worse. And so these conclusions really refer to people who've grown up in those environments. We don't have people who've been terribly deprived, and we don't have people who are in the very best possible environments. So rather than think about this as fixed, what we should think about it is that genetics really does influence our behavior if we grow up in environments that are permissive, that are good enough, and that provide us with the necessary environmental stimulation and opportunity to express our traits. The twin method for exploring these questions is really a relatively crude instrument. It gives us a sort of a rough estimate of the relative effects of heredity and environment on various psychological traits.
Oh, we don't agree on anything. We fight all the time. I mean, if she says we don't agree black, I say white. We're like sister and Eva.
It's just that principle. It's not whether we agree or not.
Clearly, the goal is to eventually get down to the point where you can look at specific genes and track their influence over developmental time.
The stupidest twin question is when you how do you know who you are when you wake up in the morning? How do you know you're you and not your sister?
So we're really, with twin studies, at a point where we can say, because of the logic of the design, the genes are important, but we really can't get in and say anything about the details.
So twin studies have not settled the age-old debate of nature versus nurture, but they have opened up fundamental questions about who we are.
What is DNA? DNA is 5 elements: carbon, phosphorus, oxygen.
Meet Joe Davis, research affiliate and resident artist at the Rich Lab at MIT. Inspired by science history, Davis is exploiting the latest genetic technology to create a unique work of art that he calls the riddle of life. It celebrates DNA, the genetic code found in all living things, discovered in 1953 by James Watson and Francis Crick.
What Watson and Crick discovered was a molecule that was ladder-like in structure, only the ladder was twisted in a helix. And in between the vertical parts of the ladder are the rungs of the ladder, and these are made of the 4 bases of DNA: cytosine, adenine, thymine, and guanine. And these are the variable parts of the molecule, and that into this variation is encoded all of the information about living things.
DNA contains 4 movable parts, an alphabet of 4 letters used in a code, the universal language of life itself.
And when scientists discovered this, they waxed a little bit poetic. In 1958, a famous expatriate German physicist turned biologist, Max Delbruck, his friend and colleague George Beadle was receiving the Nobel Prize that year in Stockholm, and Delbruck decided to play with the idea of DNA as language.
As a friendly challenge to his Nobel-winning colleague, Delbruck sent Beadle a puzzle made from 174 toothpicks. He used only 4 colors corresponding to the 4 bases of DNA. And just like in DNA, he used them in a code. He assigned different combinations of 3 toothpicks to letters of the alphabet. The model concealed a message in English.
It contained the message, which is an ancient message important to the history of art and philosophy as well as science. It said, "I am the riddle of life. Know me and you will know yourself."
Over 30 years later, Davis came across the riddle of life story while making his own attempt to encode graphics and text into DNA.
And I found that Max and George had contemplated the notion of putting information like that in a molecule, and in a way, in kind of deference to them and their great precedents, I decided to create the molecule. If I hadn't have done it, George and Max would have haunted me for the rest of my life.
While Delbruck was confined to working with toothpicks, Davis could employ modern genetic engineering. He used the same toothpick code, but worked with actual DNA to create the riddle of life molecule. His first step was to synthesize the message, broken into 4 parts.
4 parts of the 174-mer. This one may have, "I am the riddle." This one may have, "Of life, know me." This one says, "And you will know," and this one says, "Yourself." So I had to put them together in the right way. And to do that, I used these primers and a tool called polymerase chain reaction.
Polymerase chain reaction, or PCR, is one of the basic tools of genetic research. It's a high-tech copier used to duplicate useful quantities of DNA from just one correct molecule.
I inserted my little tubes, which hopefully had the correct molecules in it, in these little holes inside the PCR machine. You want to see the holes? These are immersed in a water bath, and it's alternately heated and refrigerated.
Here's how PCR works. It starts with a single strand of DNA. An enzyme helps fill it in to create a complete double-stranded molecule. Heating up that molecule then unzips the strands. The same enzyme then helps fill in the 2 single strands. That creates 2 complete molecules. Repeating the reaction then makes 4, and then 8, and then 16.
Before too long, you have billions of copies of the original molecule. And by this means, I created workable amounts of the riddle of life DNA to insert into living cells.
Davis chose living cells as the canvas for his artwork. He picked a common bacteria to carry the DNA message.
E. coli is one of the workhorses of biology because it coexists with human beings. It grows inside of us. We all have colonies, populations, significant populations of E. coli growing in our intestines. In fact, 30% of human feces is bacteria, and 10% is E. coli.
Davis uses a specially weakened strain of E. coli that can't survive outside the lab. Then, in a standard procedure, he uses calcium salts and water baths to get a few cells to take up the DNA message. Reproducing the message is now left to the E. coli.
So this is E. coli funland. We have Ferris wheels and tilt-a-whirls, and the room is maintained at the temperature of the human intestine. And E. coli's love it in here, and they have a generation every 20 minutes or so, so by tomorrow morning we'll have hundreds of billions of copies of The Riddle of Life. Now, Salvador Dali may have made 10,000 prints in his time, and Escher maybe 5,000 prints at a time, but I am by far the most successful publisher, printmaker in history, as I have now more copies of my artworks than all of the stars in the known cosmos. I can fit 10 to the 28th in a gallon milk jug. The molecule that has the information in it corresponding to the toothpicks that Max made in 1958 now exists in all of these bacteria that were transformed. The message in every one of them says, I am the riddle of life. Know me and you will know yourself.